Hello, welcome students. How are you all? My welcome to the class of biology. Dear students, Etanaga, thank you. Shillong students, please report your attendance and we'll begin right away. Alright, so we'll do a quick revision of the last chapter that we completed in the last part. Okay. Alright. Okay, thank you students. So we are beginning now. So in the last class I told you that we deal with biotechnological procedures, biotechnological studies, and that gives us a fair idea of how basically we are exploiting the use of biotechnological studies, the procedures, the examples, everything is being utilized, and we have used it to advantage to synthesize a lot of substances in our knowledge. Okay, so I'll give you a quick recap because it's been some time, I believe, so that's why, right? So we had discussed about some cloning procedures, some introduction procedures, right? So basically we are dealing with editing the gene, editing the DNA, and that is held with the knowledge of some gene editing procedures, gene cloning procedures, gene editing procedures, and one of these is basically what's the only procedure that we have is recombinant DNA technology. Okay, so we have to introduce a foreign DNA into a vector DNA. A vector is any DNA which can carry this foreign DNA or the gene of interest. You can also call it gene of interest. So this gene of interest is basically introduced in the vector, and they are cut by some restriction endonuclease enzyme. It is a type of molecular scissor. This scissor cuts the gene. This gene is introduced in the the plasmid DNA, and this becomes a recombinant DNA plasmid or a mixture of two DNA. This mixture of DNA is transferred into a carrier host, a carrier organism, and this is a transformed bacteria. It is allowed to grow and reproduce, and it is kept under aseptic sterile conditions in a growth medium, and after this it is isolated from time to time. After isolating it, we have to basically make it multiply, make it divide, make it a lot of things, and this means that we can exploit it, to use it in any way possible. But there is another problem associated with this. Although it looks easy on paper, we have to basically do it physically. We have to do in actuality. So it is not going to be as easy as it seems on paper, that you cut it, join it. It's not like cutting two pieces of paper and joining them. Basically it's so difficult because these pieces of DNA are very small. So we have to develop some techniques. If we had two pieces of paper and art and craft material or something like that, so if these pieces were very large, so we would have taken a scissor, taken a Fevicol or adhesive, join them together, place it and stick it somewhere. Okay, but this is not art and craft material. Okay, so we do not have hands, we do not have scissors. We need to have something as small as scissor at that level, which is an enzyme, molecular scissor. And then we also need to separate these parts, the large parts separated from the smaller part. So there is a technique called gel electrophoresis, which separates DNA by what size it has. Whatever is the size of the DNA, we sort it out on that basis. So this is called gel electrophoresis technique, and we separate DNA parts by the help of this method. Okay. So I explained this technique to you in the last class, that smaller fragments travel faster, larger fragments travel slower, right? And because of this, it covers a lot of distance, and in order to be able to see the DNA molecules, we want to see the DNA molecules, so we use something called ethidium bromide in order to visualize them. And ethidium bromide is used because we want to see the DNA fragments in visible light. Okay, and to visualize that, we use ultraviolet light. Okay. Cloning vector is something which is something which can increase the copies of DNA through autonomous replication. And in this we have the theory that cloning vector is selected on the basis of what is the copy number of that DNA, copy number of that plasmid. Each plasmid has its own specific copy number. Own specific copy number means it can increase the copies of its type by a particular number, and this number is fixed for each plasmid. This number is fixed for each plasmid, and we take this high copy number plasmid, introduce a gene into it, and this ensures that multiple copies of I'll send you the notes. I have given you, I have not given you this, right? Okay, I remember. I have not given you these notes in PDF form. So I'll send you these notes collectively, right? So this chapter is finished. I'm just giving you a quick recap because this is a tough chapter, I understand. So I'm just giving you a quick revision so that you can understand the difficult concepts here. And it's been 1 whole week or greater than that. So you need to revise. So copy number is such a vector which has some specific number x, y, z. So if copy number is, let's say, 15, so that plasmid will make 15 copies of itself. If I have another plasmid which has copy number 50 or 100, I will choose the plasmid with higher copy number. That means I will have higher copies of foreign DNA or gene of interest DNA also, which is mixed in that, which is introduced in the middle of that vector DNA. So 100 copies of plasmid DNA per bacterial cell means that I will get a large amount of finished product. For example, if it is insulin gene which has been inserted in the bacteria, I will have 100 copies of insulin gene. That means the rate of production will become 100 times more than some bacteria which has only one plasmid, one copy number. So that is very economically useful, right? That is the purpose. Then I told you some things. There is something called ORI. Normally we try to introduce a gene into ORI site, origin of replication. Selectable marker is something which enables us to differentiate in which bacteria have the changes been successful and in which bacteria the changes have not been successful. Why? Because I only want to choose the useful and the recombinant bacteria only. I do not want to choose the bacteria which is non-converted, non-transformed, not having the foreign DNA, not having the gene of interest. There is no use in growing such bacteria. It will be a wastage of resources. So I will choose, and for choosing I require a marker. Okay. For example, in your notes copy or even in this copy, I am selecting something, right? Selecting something means I am able to select it, highlight its importance. So I am using a marker to highlight the importance of some genes which are more useful to me, and this is called a selectable marker, right? So selectable marker is of many types. Normally we add genes for resistance to antibiotics. So if resistance to antibiotic gene is added, so if you add a particular antibiotic, all the non-useful bacteria will be killed, while the useful bacteria which have the gene of interest along with the antibiotic resistance gene, they will survive. They will survive only, right? If you introduce ampicillin resistance gene, you give a high dosage of ampicillin, all the non-useful bacteria will die, only the useful ones will survive. You can grow them in a separate culture without ampicillin, without any poison. Cloning site, something which is referring to the recognition site for restriction enzymes. So all this I have explained to you, right? But all these procedures are useful in bacteria only. When I talk about vectors for cloning genes in plants and animals, then there are separate things. I require, look, I have all these notes given to you already. So you do not need to take these notes again. You have done this in the last class, yes or no? You remember I have given you all these things in the last class. Let me get back home. I'll convert these notes into a PDF form and I will send these notes to your WhatsApp group. So this is just for your revision. For bacteria and plants and animals, what I have the problem is, in bacteria it is very easy, but normally in bacteria that means if I have to obtain some product, I will have to grow that bacteria, and that bacteria will manufacture a product and I will have to isolate it from it. But suppose I want to introduce a gene of interest permanently into some plant. Can you give me an example? Why would I want permanent resistance, permanent gene in a plant? Why should I have this property? Why not make it in bacteria and then transfer it to plant and animal? Why should I introduce a gene of interest in some plant or animal? Can you give an example where this property will be useful? I had explained you in the last class, maybe you have forgotten. If you remember, let me know. No? Okay, I will give an example. For example, genes for disease resistance, genes for pest resistance, genes for let's say resistance to environmental factors, water resistance, temperature resistance. These genes are permanently useful. You make something in bacteria, you will have to spray it again daily or something like that. But if you introduce a gene into a plant by this method, like genetic engineering, then that character will become permanent. If you introduce water resistance in a plant, this plant will now be able to survive in an atmosphere permanently where there is very less water. Other plants might not be able to grow, but this is genetically engineered plant, it will be able to grow. If you introduce gene of disease resistance, this plant will become naturally disease resistant for particular type. If you choose multiple genes for disease resistance, this plant is very useful to the farmers. Why? Because they will need to use very less pesticide and insecticide and fungicide, right? So their bills will be saved, money will be saved. If you use genes for hard plants which can tolerate mineral deficiency, so farmers will need to use very less fertilizer also, cost will be saved, right? So it's a matter of cost, efficiency. You do not need to go to the bacteria to make something and then put it back into the plant or animal, right? So this is a very, very useful feature. So that is why we are doing this. Otherwise there is no need. You make insulin in bacteria only. When you require it, you put an injection in your body. That's all. But it is not permanently useful. We are trying to go to that stage in which insulin should be manufactured in our body only to become self-sufficient. One-time treatment. Do not have to put an injection every day in your body, right? So that is why we need to move even further. In the olden times, we used only bacteria. Now we are modifying the DNA of genes of plants as well as animals by this method. So that is why we do it. So natural genetic engineer of plants is Agrobacterium tumefaciens. I told you that Agrobacterium is a natural genetic engineer for plants, and we replace the desired gene with the bacterial plasmid, and the disease causing part is removed, so it no longer causes the disease and only introduces the useful DNA into the plant or animal. Sorry, in the plant only, not the animal. Agrobacterium does not work on plant, animals, sorry. In animals, we use retrovirus. In animals, we use retrovirus. I have told you this, that for inserting DNA in animal cells, we use retrovirus. Maybe you remember now. Then we need to be able to introduce the DNA. Up till now, we have only cut the DNA. We have not introduced it inside something. We have only chosen the host We have not introduced the DNA into the host. How do we do it? We make the host so much competent, or meaning I should say, we force it to take that DNA. How do you force it to take that DNA? You need to introduce some drastic condition. Drastic condition is something like heat shock, cold shock. Heat shock, cold shock treatment is taken, and then calcium treatment. So calcium treatment, heat shock, cold shock, this causes the pores in the bacterial cell wall to become wider, and because of these pores, now the DNA can pass easily. Okay. Micro injection, take a micropipette, just like an injection, insert it into something's body. Because you are inserting into bacteria, so you require very, very thin. This micropipette is very, very costly. So I remember it costs around 1 lakh rupees for very small micropipette for bacterial cell, because it is very delicate also. You have to take a lot of care. If you break it, just try to understand, you have lost 1 lakh rupees. 1 lakh rupees, I think 4 or 5 comes, but it comes as a set because it breaks very easily and very hard to replace it also. So normally some people make it by themselves. They can have a glass factory and they learn to do it by themselves, but it is very complicated. So very expert glass blowers can do it. So normally it is very expensive. So micropipette can be used. There are some cheap ones also. They can be also used. Then there is gene gun method. You take a bullet and you take a plant cell material and you shoot it with a gun. And in this gun, instead of bullets, these are hollow point bullets, and these hollow point bullets are filled with gold and silver particles coated with DNA. Gold and silver particles coated with DNA, and this DNA basically is sprayed at high velocity on plant material. Some cells will be converted and some cells will not be converted. Lots of cells will die also, but we do not care, we have a lot of material. This is very easy method, just take a gun and shoot plant cells with it. So it's a funny way of introducing the DNA, but it is very efficient. Whenever we have a large quantity of genetic material available, we use this method only because it is very easy. Then isolation of DNA process, fragmentation process, desired DNA isolation process, joining process, transferring process, culturing process, and then extraction and scale of desired product. So all these steps I have given you. It will be a waste of time to explain all this in detail, but I think you must have got the idea of how we are doing it. Okay. So do you have any doubts regarding this chapter now? It was a tough chapter, but I believe you must have some doubts, I think. So is there any doubt regarding any step, any process, any technique that you want me to explain in detail, or detail should be explained to you again? Anything you want me to go through again? Any technique or process? Polymerase chain reaction or anything else? Is there anything you would want me to discuss again? Let me know right now. You can also ask later, but if there is anything right now, let me know. Should I move on to the next chapter? Should I move on to the next chapter? Okay. Alright, so this chapter is now complete. Biotechnologies, procedures and everything is now complete. Okay, so alright. So let us start with the, I believe you have done the applications of biotechnology and examples. In food technology and other processes, I'll mark it as a general process. I believe you have done it, right? Okay. So I'm taking a short break from the normal process here. I want you to tell me some examples of biotechnology that you already know, in where biotechnology has been very useful in preparing something. This is a break. Now I'm discussing some miscellaneous topics here, because moving on to the next chapter will be something I have to do. But before that, I need to finish this, because some questions, miscellaneous questions are asked, and that I have to cover first. So please tell me, what applications and examples do you know about, based on what you have studied or you know something? I want you to give me examples from your life, something that you know about. Actually, this is a very important part. It comes in the exam. No examples already. Everybody knows tomatoes. Everybody knows tomatoes, right? So, tell me, have you heard about flavor saver tomato? Have you heard about flavor saver tomato? Yes or no? Yes? Red cabbage, potato, and thick skin tomato. Alright. Actually, flavor saver tomato is very important. Not yet, okay. This comes in your exam, so I'll have to explain this. Look, flavor saver tomato is a type of tomato which is normally it's coming less these days, but it's a type of tomato in market that I want to tell you about. Please write about it. It was the first commercially grown, genetically engineered genetically engineered food to be given license for human consumption. So, it had an enzyme called polygalacturonase, and okay, just a second. And why was it so important? This tomato, you can keep it in outside the fridge for 1 month and still it will not get spoiled. You can keep this tomato outside the fridge in non-refrigerated condition and still it will not get spoiled for 1 month. Do you understand the importance of this? This means that farmers will lose less money growing tomatoes, because tomato degenerates very rapidly. Once it is ripened, it will very quickly undergo degradation, and there is nothing else, no option except preparing tomato sauce from it. Spoiled tomato, if tomato is getting spoiled, you should rapidly wash it and make it into tomato sauce or something, otherwise it is gone forever, right? So what is that thing which makes tomato get spoiled so easily, right? This is not given in NCERT, still question is coming. That is why I am teaching this. So there are normal tomatoes contain an enzyme called polygalacturonase, which is found in ripening fruit. Ripening fruit. It is responsible for softening and spoiling of tomato fruit. So what do? We destroy this enzyme. How do you propose that we destroy this enzyme? Do you have any method? I want you to use your brains and try to tell me a method how you will destroy this enzyme. Can you propose any method based on the chapter that you have just studied? Is there any way to do it? Based on the chapters that you have just studied. I should better put it this way. Based on the previous chapters, can you propose a way in which I can destroy this enzyme? Let me give you a hint. Can you propose any way in which I can prevent this enzyme from ever being formed? Because if it is formed, the fruit is spoiled. I do not want not a single percentage, not even 0.01%. I don't want this enzyme at all. What should I do? Use your brains. Try to use your deductive power. What should I do? Question is asked. Okay, please mark this enzyme, very important. It is really important, that's why I am marking it. Genome editing. Yeah, it's good. What part of genome will you edit? My question changes. What part of genome will you edit? And what exactly do you mean by editing? Do you want to delete something? Do you want to add something? Or do you want to subtract something? What exactly do you want to edit? Editing means adding or subtracting. So what are you going to subtract? What are you going to delete? Replace. Okay. Alright. Just like video editing, you are replacing some video clip with something else. What do you want to replace it with? And how do you propose we do that? Yeah, you're going in the right direction. but I need a more specific answer. Okay, all right, that will do. I understand that you got an idea. We will not replace it, we will delete it. We won't delete it either. We will not delete it either. We will prevent it from ever being formed. And how do you do that? You do it by antisense RNA technology. Are you familiar with gene silencing? Do you remember gene silencing? Yeah, molecular scissor is a good method, but this gene is still hard to cut. We normally try to do it, but the problem becomes that identifying this gene is very difficult, and when we try to cut it, we will actually have to replace some useful genes as well. So we do not cut it. We have another technique, and that is called gene silencing. You remember gene silencing, what it is? Yes? You remember gene silencing? Say yes or no. If you remember it, then I will proceed. Okay, all right. You do not remember. Okay. Do you remember RNA? Do you remember that I taught you RNA? Do you remember RNA? Do you remember me teaching you RNA? Say yes or no. All right. When I taught you RNA, then I told you that at the time of translation, when I taught you RNA, after that I taught you translation. Try to remember protein translation process. Polygalacturonase is an enzyme. If it is an enzyme, then that means it is a protein. If it is a protein, then that means it must have been made by some messenger RNA. This messenger RNA is coming from some DNA, right? So there is a step I told you that when RNA is formed, it is formed from only one strand of DNA. Do you remember? RNA is formed only from one strand of DNA. If you remember this, please tell me what is that name of DNA strand called? What is the name of that DNA strand which makes this RNA? Is it called coding strand, or is it called anti-coding strand or non-coding strand? I am waiting for your answers. I want you people to exercise your brain. Use your memory. Yeah, very good. Anti-coding strand. And then I told you that DNA's anti-coding strand makes RNA, right? Non-coding strand. It actually makes the RNA from which this RNA is called hnRNA. hnRNA is converted into messenger RNA by splicing and capping and tailing. Now you remember everything? Okay. So messenger RNA is coming from anti-coding strand, but coding strand is not used. If you make something from coding strand, then that is called antisense RNA. Okay, if you make RNA from coding strand, then that means you are making something which is complementary to the already formed messenger RNA. This other RNA called antisense RNA will stick to the normal RNA, and normally RNA is single stranded, but if you make RNA from other DNA strand, this new RNA will stick to the messenger RNA, forming double stranded messenger RNA. This is a waste of everything. Now messenger RNA will never be used in making protein. This is called gene silencing, gene interference. Now you remember this technique? Say yes or no. I told you that there was a Nobel Prize given for this technique. It is called gene interference, RNA interference technology, and it is used in many processes. If you remember, say yes. If you don't remember, still say no. Then I'll explain again. Yeah, maybe you were absent, but this is that technology that I am telling you about. Normally, you make RNA, messenger RNA from one strand. Other strand is not touched at all, but if you make something by, you take the other strand of DNA, make some RNA from it. Okay, how do you do it? You take this DNA strand, cut it. We will edit this gene, but we will edit this gene in such a way that it is placed somewhere else, so that it can be, because this place you can, at the same place you cannot make the other RNA also. Normally all genes, whenever they are made, whenever the gene is being read by the RNA polymerase, it will read only one strand of DNA. It will not read the other strand, whatever you might do. So what we can do is, we can cut this other strand, non-coding strand, and put it somewhere else in the path of some other enzyme, in the path of some other gene. There it will be read, and there it will form a new messenger RNA. This messenger RNA is antisense RNA. It will interfere with that other gene where the single stranded RNA is formed. Okay, so you have to make double stranded RNA. This phenomenon is called gene silencing. You can silence any gene by the help of this technology. And for this technique, there were 3 scientists. Main work is of 2 scientists. They were given the Nobel Prize for it. It is called RNA interference technology or antisense RNA technology, and this is how you do it. In this polygalacturonase enzyme, we apply this technique. So please write. This enzyme is silenced the antisense RNA is used to silence the expression of polygalacturonase enzyme. In simple words, used to silence the polygalacturonase gene. Once you silence it, it will not be used. These tomatoes the gene becomes deactivated. Okay, I'll write this statement first, then I'll move on to the next page. The deactivated gene no longer produces the enzyme, no longer produces the enzyme, thus increasing the shelf life of tomatoes. Even without freezing, even without refrigerator. So just like apples and mangoes and oranges, you can keep it in wooden boxes without freezing. Without refrigeration you can keep, and they will not be spoiled. In market, you normally call it as a hybrid tomato, but hybrid tomatoes are of many types. Flavor saver tomatoes, one type of hybrid tomato, not necessarily in your market, local market. Okay. There are some other GMO crops as well. Okay, so this is done. Then there is something else called golden rice. Golden rice. And golden rice, there is a question from golden rice in NEET 2018. Sorry, NEET 2019. You remember the question from golden rice? Why do you call it golden rice? Why do you call it golden rice? And what is the reason? Color. Yeah, there is the color, but this color is having something else also. Okay, so I'll explain. Alright, so I'll explain what is happening. Before I go into this detail, let me tell you what happens. Do you know what thing is responsible for the color of carrots? Do you know what thing is responsible for the color of carrots? Color of carrots. What is the substance, chemical substance? The name of the pigment? Carotene. Okay. But can you tell me, does carrot contain vitamin A or not? Do carrots contain vitamin A or not? You must have heard Bugs Bunny, and there are so many cartoons which always keep eating carrots. So can you tell me, what is the thing that is responsible for the color of carrots? And do carrots contain vitamin A or not? It is connected to golden rice in some way. I'll explain once I give you this example. Carrots do not contain vitamin A. Carrots do not contain vitamin A. Now you will get confused. Why is sir saying that carrots do not contain vitamin A? Right? Carrots actually do not contain vitamin A. They contain something else. They contain beta carotene. And beta carotene, there is only a very slight chemical reaction, a slight oxidation reaction. It is quickly converted into vitamin A in our bodies. So actually carrots do not contain vitamin A, but they contain precursor of vitamin A, which is this bright orange, yellow colored compound. Now you understand? Yeah. If you say to anybody, if you tell anybody, your teachers also, anybody, you say that carrots do not contain vitamin A at all, they will be surprised. They will say that you are thinking wrong. This is not what's true. But actually, that is the case. Carrots do not contain vitamin A, they contain beta carotene. And this is a very quick reaction. It happens very rapidly in our body, not in the body of carrots, not in plant body. It happens in our body. the carrot pigment is rapidly converted into vitamin A, which is a very vital pigment for our vision. Okay, rods and cones, basically we use a visual rhodopsin pigment, and for that we require vitamin A, and deficiency of vitamin A causes night blindness. So what I am trying to tell you is that instead of eating carrot, you can eat something else. Do you know marigold flowers? Do you know marigold flowers? Marigold flowers. What color is marigold flower? What color is marigold flower? Marigold flower, let me tell you. Okay, give me a second. Yeah, marigold flower is something like this in color, right? Dark yellow, orange, something like this, right? So basically like this, and this is the same color of carrots. So instead of eating a carrot, you can eat a marigold flower, and you will get the same amount of vitamin A. Carrots have the same pigment as marigold or a different flower, and that flower is called daffodils. Have you heard of the poem called Daffodils? Have you read this poem? Daffodils. Okay, wrong color. Yeah. Daffodils, do you know? Nah. Okay. Nobody recognizes daffodils. All right. Okay, all right. Give me a second. Well, actually, I wanted to show you some daffodils. Daffodils not exactly like this. I have like the thing I have drawn. There is a poem. I think it was by it's a long time since I have read it. It's by William Wordsworth. And why I'm telling you all these things that I have depicted here, all these things that I have drawn here, they have beta carotene. Beta carotene is found in many flowers. Beta carotene, it is found in many flowers. Okay. Many flowers, carrots, etc. So the theory behind golden rice is that in many countries such as India, Africa, Malaysia, Indonesia, and basically Southeast Asian countries, these people, basically Southeast Asian people, China, Mongolia, Japan, they eat a lot of rice, right? And so they assumed, let me put this word, they assumed that these people are malnutrition. Indians are always supposed to be malnutrition. I know that I eat very healthy food in my home, but normally people assume that we are malnutrition because World Health Organization has put up some reports which say that basically in India people are having very less nutrition, particularly poor people. So what do you want to do about it? So it was not properly studied, but there was a lot of debate about it, that Indians don't need GMO crops like this. But there was a company, Monsanto and some other companies, what they did was, they decided to make a rice which is yellow in color, which has some beta carotene. So basically when you eat this rice, you will get vitamin A directly. You do not need to eat carrots, you do not need to eat flowers. Yeah, that's a joke. You will get vitamin A whenever you eat your rice. So basically you can assume that Indians don't eat salad or anything else. So if you eat this, this will be able to cure malnutrition in large parts of the world. So this was not successful. Nobody liked the taste. It's very awful tasting, and because the taste is not good, and it's also very expensive. So this was a total failure. But CBSE loves this question. They ask it every year, and this was asked in NEET 2019 exam, and this was a very particular question. Nobody knew. Okay, only some very conceptual minded people knew the answer. Thing is, daffodil, basically daffodil or marigold, any other flower, they all have yellow orange colored pigments. They all have yellow orange colored pigments, and it can be used to make vitamin A. So actually golden rice does not contain vitamin A, but it is marketed as vitamin A enriched rice. And the question in CBSE exam was, NEET exam was, where do you obtain the gene for making this carotene in golden rice? Do you know the answer? Do you know the answer where the gene for carotene in golden rice was obtained from? Yes? Nobody? Okay, I'll write the answer. To cure malnutrition in many countries, malnutrition in many countries, in many Southeast Asian countries, golden rice was developed. Vitamin A enriched rice. It is called vitamin A enriched rice, and the question is, where do you obtain this? So I am telling you the answer. It was created, golden rice was created by transforming rice is Oryza sativa with 2 beta carotene biosynthesis genes from daffodil. So that is why I was saying daffodil. Nobody answered. You could say marigold, you could say carrot, you could say daffodil. So this answer was hidden, just like an Easter egg. Okay. And there is also a soil bacteria. And daffodil and some number 1 daffodil and number 2 from soil bacteria. Why I am telling you this soil bacteria? Because in NEET 2019 question came from daffodil. In NEET 2020 or 21, I am not sure which year, most probably this year, they can also ask that there were 2 genes, where is the other gene obtained from? So 2 biosynthesis genes, first was obtained from daffodil, the other gene was obtained from Erwinia uridovora. Just need to learn this. So basically it makes beta carotene. Beta carotene can be obtained from anything which is yellow in color or orange in color. So you can have basically a yellow colored capsicum, you can eat a yellow red colored chilies, you can eat yellow red colored papaya, you can eat mangoes, you can eat anything which is golden colored, yellow colored, orange colored. So you can eat an orange also. So everything that is of this color that contains beta carotene. You can eat flower. When you eat a marigold flower petals, they will also have beta carotene, and when it goes into your body, you will make vitamin A from it. Erwinia, it's W. I'll write it again. Erwinia uridovora. Okay. So this is the science behind golden rice. Look, 2 questions are asked, very common. Golden rice is, you can see the past year papers. It's a very common question. That is why I am discussing it with you. So it's not given in NCERT. That is the problem. They ask some questions which are not given in NCERT at all. BT cotton is given in NCERT, but golden rice is not properly discussed. Not even this information is given, what I am telling you right now. But the question came from, he was asking about which is the source of this gene. So daffodil is the source of that gene. Daffodils only those people know who have heard the poem called Daffodils by William Wordsworth. You can search it on Google. Daffodil by William Wordsworth. He was an English poet, and I think he was in 16th century or 17th century, and he used to write a lot of poems. He was a well-known English poet. Daffodils are yellow color. Okay. So from that they obtained golden rice. Okay. Finally, so this is over. Okay. Alright, so for a few more genetically enhanced crops I want to discuss with you. Let us discuss them. Alfalfa, you know, BT cotton, you know, I guess you know something else also. Maize, soya bean, sugar beet. All right. I'll give you some example. Some genetically modified crops grown around the world. Genetically modified crops grown around the world. So the maximum cultivated genetically modified crops as of now, it is genetically modified soya bean. Soya bean comprises 50% of all the okay, wait a second, I'll have to make it. Yeah, okay. I'll make it by hand. If I make a chart, then in this chart, if I talk about soya bean makes 50% of all GMO crops around the world. This is for soya bean. Genetically modified soya bean. Genetically modified soya bean. Then there is something else called genetically modified maize. This is about 30% genetically modified maize. This is 30%. 50% is soya bean. Then genetically modified cotton. Genetically modified cotton. It is about 14%. Then there is a small percentage of genetically modified canola. This is 5%. 1% for others. So basically you can get an idea that the maximum cultivated GMO crop, genetically modified crop, it is soya bean. After this maize, after that cotton, after that canola. This is just to give you an idea. Beyond this, there are other GMO crops, and that will be discussed with you in the chapter, that is biotechnology in human welfare, food crops, etc. All those varieties of food crops, that will be discussed later. So these two things are very, very important. I'll stress on it again and again. Golden rice is very important. So it comes time and again in the examination. Okay. So other, if you want to know the names of other crops, alfalfa, soybean, squash, GM, genetically modified papaya, and there is one thing else called golden apple or Arctic apple. So golden apple, Arctic apple, all these are GMO crops, genetically modified crops. Okay. So I'll give you the list from time to time. I think this goes enough for today, right? Alright, so let's do something else in about whatever part we have left. Is there anything else you would want to ask me about it? Okay. Let me give you some examples of other food crops that have been asked. Miscellaneous, today's topic is miscellaneous questions that has been asked. So one question has been asked about bromato. What is bromato? It's a hybrid crop of brinjal and tomato. Look, these are very silly questions, short questions. There is no logic in explaining the full details. Just learn it. What is bromato? Okay. Hybrid of brinjal and tomato. What is Raphano brassica? Raphano brassica. Do you know? Okay, you don't know, I think. Look, there was a scientist called Karpichenko. Yeah, canola is a seed from which canola seed oil is prepared. What is canola, you are asking me? Canola is basically used to prepare a type of seed called canola seed oil. Are you aware of canola seed oil, yes or no? I don't think you are. So let me tell you about it. Okay. How many people of you are aware of mustard oil? It's a silly question to ask, but have you ever cooked with mustard oil? Look, I normally cook my food in mustard oil only, so I'm well aware of its properties. Yeah, okay, I can see a lot of hands there. So everybody knows what mustard oil is, right? So let me explain what happens is, when you cook food in mustard oil, there is a problem. Mustard oil gives a lot of pungent odor. Actually, that is very useful for you because mustard oil is anti-cancerous, anti-diabetic, and it is also helpful in reducing the obesity. Okay, so I eat a lot. I eat a really lot of things, and I'm not as obese as I should be. Okay, so maybe that is because of mustard oil. All right. So I have recently gone full overboard with a lot of food. I should not have, maybe. So that is because my mother cooks very good food, and that's one reason. Another reason is depression. I'm partly depressed because of some events that happened about 2-3 years ago regarding my breakup and all. Anyway, so what happens is, mustard oil is having a very pungent odor, and some people cannot tolerate that odor, that burning in the eyes and all that. Actually, that is good because of a glycoside, but some people cannot tolerate it. So there is another plant which is similar to mustard, and it's called rapeseed. Rapeseed is basically mustard only, and this variety is called canola. Canola is having a special kind of, it's a seed similar to mustard, belongs to mustard family, Brassicaceae family. But this rapeseed produces an oil called canola seed oil, which is having very less quantity of erucic acid. So when you cook food with it, there will be no pungent odor. You can cook in it and your eyes will not keep watering and you will not be very dissatisfied with the odor of this. So it's the pungent odor is highly reduced in canola seed oil. So that is why people are switching from mustard to canola. Okay, that is the reason. The name of canola botanical name is Brassica napus. Mustard is Brassica campestris. Canola is Brassica napus. The flower is exactly similar to mustard flower. You cannot tell the difference. So basically we have a small difference, but that is not easily visible. That is the only thing. Anyway, so I was explaining to you what Raphanobbrassica is. So Karpichenko was a very brilliant Russian scientist. Very brilliant. I'll tell you later why I'm calling him brilliant. He wanted to basically exploit nature in some way. So he was observing a lot of hybrids. He wanted to make a lot of hybrids. So he saw mule. Mule is a cross between horse and donkey. So he said that let me make some hybrids in plants. And he said that look at Brassica capitata, cabbage. Cabbage is having some part which is above the ground and it is edible in nature. Then there is Raphanus sativus. Raphanus sativus, everybody knows what it is. Raphanus sativus. Everybody knows Raphanus sativus. Mooli. Yeah, radish. Everybody knows. Thank you. So radish is having, most people do not eat radish leaves. Normally in North India we make green type of saag out of it. I'm not sure you are aware of it. So saag is made and using Raphanus sativus leaves, but I'm not sure everybody knows about it. Anyway, Karpichenko said that there is something which is underground in radish and it is edible and we do not eat the leaves. And in cabbage, we do not eat the root, we eat the upper part. So let me make a hybrid in which I will get cabbage above the ground and radish below the ground. So this was his brilliant idea. So he made a hybrid out of cabbage and radish. But what the end result was, he got a plant which had leaves like radish but root like cabbage. Okay, so this was a failed experiment. He was trying to make something which had edible part like radish underground and edible part above ground like cabbage. But what he got was cabbage like root, inedible, and radish like leaves. Totally, utterly useless plant. So that is why I'm calling him brilliant. Yeah. So Karpichenko produced a hybrid. So this was the first plant hybrid, artificial hybrid or man-made hybrid between Raphanus sativus and Brassica capitata. Brassica capitata, a variety. Okay, basically, sorry, Brassica oleracea variety capitata. Oleracea variety capitata. This is cabbage. This is radish. So you will not get the very detailed question. Question was, what is Raphano brassica? It is a cross between what plants? Which plants were crossed in making Raphano brassica? So Raphanus sativus, you know, brassica has many species. Brassica oleracea variety capitata, cabbage. Brassica oleracea variety gongyloides. It is something called, we call as a knoll hole. Brassica oleracea has a third variety also, okay, and that is cauliflower. It is not capitata. So 3 varieties. So the question becomes very, very confusing. Which variety? So that is why I am writing the variety. Okay. So question will not become confusing after this. So let us take this down for today. We covered some miscellaneous questions and topics. So please just keep on adding this somewhere, and from the next class we will finish the remaining parts of class 12 syllabus. Okay. So I felt this was very important because this comes in exams every year. So I'll give you this also. You can ask me for the notes as well. Okay, and if you have any doubts regarding this or anything, let me know. So thank you, dear students. Travel back home safely and good night. Arigatou gozaimasu. Thank you.